Hi, I'm Sandra Porter, and I'm coming to you live this week from Seattle, Washington. This week, we're going to be comparing the structures of DNA and proteins. The proteins that we're going to look at are a special group of proteins called restriction enzymes. Restriction enzymes function by cutting the phosphodiester bonds that hold the nucleotides together in strands of DNA. We're going to look at how the restriction enzymes interact with the DNA, and we're going to spend time looking at all the chemical properties of the amino acids that make up proteins. To people who work in bioinformatics and molecular biology, the chemical properties of the 20-some amino acids are kind of like the alphabet. They help us understand how sequences differ, what parts of sequences are important, what differences might be important, and give us insights into the functions of the proteins. In this segment, we're going to talk about the kinds of information we get when we search the molecular modeling database at the NCBI. I begin by entering the name of my structure, which in this case is 2BAM. Then I click Search. This searches all the NCBI databases at the same time. And you can see there are several here in this diagram. The numbers that appear to the left of the icons for the databases reflect the number of entries for that search item within that database. So in this case, I see there are 3 entries in the nucleotide database for my item 2BAM. I see there are entries in the protein database and entries in the structure database. Now I want to see the information in the structure database, so I select this link. The first thing that I see is the lists of the records that match my query. If I had multiple records that matched, I would see a short description for each one. In this description, I see a little graphic that shows the protein. I see a title for the entry in the database. I see some information about taxonomy. I see that this entry has 2 proteins, 2 nucleic acids, in this case DNA, and 1 other kind of chemical, and it was last modified, looks like July 19th this year. I also have links so that I could work with this information directly, but we're going to select the title right now and look at the entire structure record. So the structure record has considerably more information about the structure. It has a title. It has titles of citations. These are publications in peer-reviewed journals that have some information about our structure, and I could look at this one, or I could select the link to see all the references that go to this. For each one of these, I have a title of an article, and I could select the title, and I could get an abstract that I could use to read more and learn more about this enzyme. I also see some information about biological units. This one says it's the first biological unit, but we have other information about this. We also see whether or not there are asymmetric units. And notice that we also have little question mark icons here that we can use to get more information. And I will leave it to you to select that question mark and learn more about the biological units and asymmetric units. Other information that we see on this page concerns the PDB ID, which we've been using, 2BAM. We have information about when this structure was first deposited in the database. We can see here that was in August 1998. We also see some information about taxonomy. But remember, when we looked at the other page, we saw that there was more to it than a synthetic construct. And if we click this arrow and open up the taxonomy information, we see that molecules in the structure actually come from different sources. We see that chains A and B, which are proteins, as we'll find out in a minute, come from Bacillus amyloliquefaciens, which is a bacteria. And we see that chains C and D are synthetic constructs. In fact, those are short pieces of DNA that were made by a DNA synthesizer. We see a little bit more information here as well. We're going to cover VAST in a little bit later, but we see that this structure was solved by X-ray diffraction, which is a common way of finding out information about structures. And we could click the question mark to learn more about it. Continuing on, we see a diagram that shows us how some of the molecules in the structure interact. We can see that the two protein chains are interacting with each other, as well as with the DNA chains, and the DNA chains are interacting with each other also. If we wanted to download the structure, we could download it by selecting View Structure. Continuing down the page, we see that there is a key to tell us which items are proteins, nucleic acids. I don't know why they used nucleotide in here. It actually should be nucleic acids because these are more than one nucleotide. And they use the diamond shape to represent other chemicals. And as we look here, we can see that we have two protein chains, A and B, and two nucleotide chains, C and D. And we have two calcium ions as well. The links here in the chemicals part of the table take us to a database called PubChem, where we could learn more about any of the chemicals that were listed in this section. The other information we want to know about here concerns our protein. Now our protein's name is endonuclease BAMH1, and I'm going to scroll up to the top here and point something out about that. Here it says BAMHI. It really should be a 1, BAMH1. I think since many times people use, it's by convention, people use a Roman numeral to represent the number in which an enzyme was discovered. I think that may have confused the people who designed the database, and so they put in I's instead. But it should be BAMH1. As we continue down, back to our table, we can see there's a link here. It says Show Sequence Annotation underneath a graph that represents our protein chain. And if I select that, what I can find is the length of our protein. And I can see here that both of my protein chains are the same length, because I only see one graph. And I see that this is 213, we'll zoom in a little bit. I see this is 213 amino acids long. I also see that there's a section of this protein from amino acid 1 until amino acid, I think this is probably about 180 or so, that belongs to something called the BAMH1 superfamily. A superfamily is a group of proteins that are related by evolution. So these sequences would be very similar to each other. And if we wanted to take a look, we could click that, and it would take us to a page where it would show us something about these conserved domains. And we could see that here are different members of the superfamily. We could see where the amino acid sequences are similar to each other and where they're different. We will be discussing the molecular modeling database in further detail at a later date.